Okay, so if you could introduce yourselves for us.
Yeah, hi everybody. So my name is Alan Liu, and I'm a associate professor in mechanical engineering and biomedical engineering at the University of Michigan.
And I invited you today because I read your paper on synthetic biology and biomaterials, and I thought it was really interesting. So I'm hoping that you can share a little bit about that with us.
Yeah, absolutely. This is I'm glad you invited me for this podcast, and this was a perspective article that was co-authored by 20 plus authors, PIs, all of them. It began actually with a discussion that took place in the fall of 2019 before the pandemic started. I was actually chairing a NSF workshop on Square Table, workshop on biomaterials and synthetic biology, and invited about 40 PIs to that meeting. And this was a very interesting meeting that the NSF wanted to focus on this two particular topic. And we brought together half the group was synthetic biologists, half the group were biomaterial scientists. And we had 2 days of stimulating discussions around multiple topics. And at the end of that workshop, we decided that it would be nice to write a perspective article together, but took a long time to get that going, because of the pandemic and other, demands come up. But we're very happy to put that perspective article out earlier last month. So it was great.
Mm-hmm. Yeah. So I really appreciated it was such a sort of comprehensive breakdown of the field and how the two can overlap and link into each other. Yeah, so I thought a good place to start might be to sort of talk about what are biomaterials, or what are we actually referring to when we say the word biomaterials.
Sure. Yeah, so I think biomaterials can take on different type of definitions, and one way to think about, well, some of the times we think about biomaterials as natural biomaterials, right? So they can be materials that are synthesized by bacteria, or they can be protein-based biomaterials that we find in our body, like collagen, fibrin, or they can be polysaccharide-based biomaterial like agarose, alginate, gel, so on. But I think in the perspective itself, I'll talk about some of the advances in biomaterials. We tend to think about biomaterials as referring to a substance that has been engineered as part of a complex system that can control the interactions with components of a living system, right? And the goal is to be able to direct cellular functions. And one area of biomaterial research that's very has been sort of rapidly progressing for many years now is to use these materials to direct stem cell fate, right? So to direct the differentiation of cells. So that's sort of the working definition we'll go with for biomaterials here. I also want to share a quote from Dr. Mina Basel, who is a pioneer in the study of extracellular matrix, or ECM, on cell function. And her quote sums it up very well, right? That half of the secret of the cells are actually outside the cell. So the materials that live outside of living cells are actually very important for understanding cell function and physiology.
Yeah, that's an important point, actually. So you would say that although there are, say, cellulose and agarose and those sorts of things, biomaterials would, I guess, more be focused on the actual cells and manipulating cells to be material-like, or be in materials, would you say?
I would say be in material, yeah. So you can imagine cells sitting embedded in a matrix, and that material can actually guide the cell function, right? So one of the areas people have done a lot of work on is to build synthetic extracellular matrix materials. So these are not the natural biomaterials that we find in our body, but these will be synthetic polymers that will present ligands for the cells to interpret that cues, and then be able to sort of govern their cell function that way. So that's sort of the framework of the biomaterials and cell interactions that was referring to.
Okay, yeah. What would you say are, I guess, sort of the biggest advancements in biomaterials at the moment that are sort of, yeah, I guess that's my question.
Yeah, sure. So let me highlight a couple areas here. So we're just talking about cell biomaterial interface, and a major focus of that area is in the development of hydrogels. So these hydrogels are basically water-swollen polymers network. And these hydrogels can be formed by a variety of natural or synthetic polymers. They have very, very important biomedical applications in drug delivery, tissue engineering, or regenerative medicine. So one area of biomaterial research that is trying to recapitulate this complex mechanical behaviors in natural extracellular matrix materials or extracellular matrix. So it turns out that the natural ECMs in our body or living tissues are not linear elastic materials, but rather they will exhibit nonlinear viscoelastic behaviors. So in order to achieve that with sort of the I guess, synthetic ECM, you can incorporate dynamic or reversible cross-linking interactions, and instead of using static covalent interactions. So this will allow one to change not just the stiffness of the materials, but also the viscoelasticity of it. And it's been shown that this viscoelasticity in these, in these hydrogels can have profound impact on cell processes like proliferation, apoptosis, migration, and differentiation. So that's sort of one aspect of I guess biomaterials.
What sort of ECM molecules are you working with when you talk about sort of cross-linking and those sorts of things? What molecules?
Yeah. So what people have used, include alginate gels. So polyacrylamide is another gel that people use, but that's an elastic type of network, and people also use collagen gels in that, or peg gels, and these are sort of common sort of synthetic polymers that people use. And material scientists would use different molecular weight, different cross-linking, and then try to recapitulate some of these material properties of gels. And then what they would do is try to decorate them with cell adhesive molecules so that cells can be embedded or live in it, right? Yeah.
Right. Yeah. Are there sort of applications of these hydrogels out there now, or is it sort of still in the research phase?
Oh, I think, I think there are probably some out there in terms of developing, so the area like regenerative medicine, building tissues or drug delivery. I think there are some polymeric materials out there that was, probably are in the sort of, clinical stage of being tested and whatnot. And certainly I think they're out there, not sort of the most familiar with, where some of these products are at the commercial stage, but I'm sure lots of them are in going in that direction. Yeah.
Sure.
I would also highlight, just since we're talking about the biomaterials advancement, the other major area is to make these biomaterial to be responsive to stimuli, so to be dynamic and responsive to stimuli. And the stimuli can be physical, like temperature, pH, or force, or light. And then they can also be sensitive to other biomolecules, like chemical cues like DNA, proteins, or small molecules. I think that's another major area of research in to allow these biomaterials to be responsive to particular stimuli, so they can be
So in your paper you spoke a lot about sort of these materials being programmable. Is that sort of what you meant by programmable? Is there something else, something more than those?
Definitely. So the programmable aspect, so this is one way of thinking about programmability in the material itself is by having molecular moiety that can so think about input output, right? If we put in something that's temperature sensitive, we can change the chemistry, or something that is pH sensitive, or something that is sensitive to binding to small ligands. So that's a programmable aspect of biomaterials. The other programmable piece is actually in the synthetic biology part, that we can, and we'll probably talk about this later in sort of the engineer living materials, is to engineer living cells that will then be, which is programmable, right? This is the whole premise of synthetic biology, and that programmable cells, that program cells can interact with the materials, and that's another aspect of that programmable piece. Yeah.
So I'm actually curious about where you would say this kind of the sort of approach of synthetic biology sort of begins in creating programmable materials? Because at the moment, it seems to me like there's like what you were talking about before with the, you know, like cells responding to different sort of pHs, etc. Wouldn't that be a synthetic biology approach, or where does it start?
Yeah, that's a very good question. So, you know, so in that context of embedding cells in a gel, like if the cells have never been manipulated, right? That just basically culturing a cell in a gel to see how the gel may, or this biomaterial, this extracellular matrix will impact cell behavior. And that, to me, that is not, there's no strong elements in that biology there. Whereas if you start taking a cell and engineer that cell to be, you know, to be different, right? To have other behaviors, then you can have this bidirectional information transfer between the gel and the cell back to the gel, or the cell back to the environment, right? So I think that's when I would sort of see the cross-fertilization between biomaterials and synthetic biology, at least in that context, right? If you're simply just embedding a cell in a gel, that I think most people would agree that is not synthetic biology, right? Yeah.
So you spoke about sort of the concept of living materials. Could you explain to our viewers a little bit more about what living and engineered materials are?
Yeah. So this is a central aspect of thinking about what you know, biomaterials and synthetic biology can, you know, when you put the two together, what can come out of it. And I think, you know, in you know, engineering, you know, living material essentially implies having cells in a material, and that the engineer part is where the synthetic biology comes in. That if you can engineer cells that would have certain behavior, certain you know, sensing abilities, then that essentially that material will have the ability to do something different, right? So one example of this, or be able to live, just like how sort of some of the natural materials in our, you know, nature are, right? Basically, you have co-culture of, you know, bacteria and fungus, and they do interesting things. And so one example of that, you know, maybe will help to bring this idea out more is bioconcrete. So bioconcrete is using microbiologically induced calcite precipitation, and you can mix sand and hydrogels with bacteria into, you know, and the bacteria will actually grow in this material. The bacteria will basically reproduce in the medium in that material, and actually biomineralize with calcium carbonate that's contribute to strength and durability of the concrete. So this type of material has actually been developed already, and this, you know, have the, you know, potential way of having this self-replicating concrete, and can be used in, you know, a variety of applications. So this, you know, is essentially a good example of embedding synthetic biology with a material. And there are other examples that we can, you know, sort of talk about later on if there's a, yeah, if there's an interest. Yeah.
Just going back slightly to the sort of programmable materials, could you give an example of one, sort of how it works mechanistically? I mean, I'm not a material scientist, so maybe not too in depth, but a little bit.
Yeah. Yeah. So neither am I, but if I go back to the sort of, I guess, the programmable in the stimuli response case, there are some molecule, you know, so there's some pH sensitive moiety that can be put in, for instance, that would change the conformation of the polymer. Like you can actually make, you know, or polymers that can be degraded. So by engineering a protease cut site into the synthetic polymer, so that when cells are embedded in there, let's say secrete a protease, and that protease will cut the gel up. So that's another sort of programmable piece in there. Like if you have a cell that's becoming malignant that will secrete protease, then the gel becomes, you know, degraded, right? So there's types of biomaterials out there that basically are sort of based on this type of concept of having a stimuli or light responsive. I think that's the easier one that many people actually do use light sensitive domains that you know, can be cross-linked or upon light illumination, so you can change the stiffness of the gel, like in situ, right? So yeah, and it can pattern that as well, right?
And what would you say are some of these sort of light controlled materials? What are some examples of the light controlled materials for specific applications?
Yeah, for specific application, yeah, the ones that come to mind are probably not so application based, but more for sort of basic studies. People are interested in looking at how cells migrate on a substrate with different stiffnesses, and if you have a gel that can be stiffened based on light elimination, and you can have a cell that's migrating toward randomly or toward particular locations, and upon light elimination, this gel becomes stiffer, and the cells will no longer, there's this idea of durotaxis. The cells will actually migrate to a stiffer area, so you can use it as a way to guide cells. I'm not sure what practical application will be for a light base, at least in vivo. It'd be hard to imagine because you have to use a different type of wavelength. Other type of stimuli responsive materials could be using ultrasound. I think that's another great physical cues that is actually very useful for tuning biological responses. But in terms of materials, I mean, there may be some acoustically activated gel materials out there. I'm not sure myself. Yeah.
Exciting stuff. Sounds pretty cool.
Yeah.
I think in your paper you also mentioned multiphase biomaterials. Could you talk a little bit about that?
Oh yeah. So that one has to do with this new field, or this nascent field of liquid phase separation. And essentially, there are liquids that, as we know, liquids will basically mix together that into a uniform solutions. But it turns out that in biology we have liquid that will basically not mix together, right? So form these phases. So the idea there is that we can create materials that have multiple components that basically was segregate, I guess, sort of have form these different phases, and that's sort of the concept behind it, I guess. Yeah. Okay. I think we also talk about like DNA, other macromolecules that that could be used in this sort of multi-phase material, right? Yeah.
Sure. Yeah. So would that be like, sort of talking about it, saying like, this is how synthetic biology can be in these cells. We've already done it with these sort of cells and these multi-phase materials, so it's not far removed that we can engineer these cells and put them in there.
Yeah, I think the multi-phase material part was more of a biomaterials advance, or from a biomaterials angle.
Right.
There's, I guess, these phase separated condensate, biomolecular condensate, are also thought to be ways of organizing reactions. So proteins or certain nucleic acid would basically form a condensate, and that is a way to organize reaction within the cell. I'm sure there are some folks are trying to engineer that within, let's say, bacteria to organize different reaction within that. But I'm not sure, how easy is to sort of think about the connection between, synthetic biology and biomaterials in that particular, from that particular angle. Yeah.
Right. Yeah. That was called a condensate, did you say? Yeah,
condensates. Yeah, biomolecular condensate. So, and basically certain proteins, intrinsically disordered proteins with RNA molecule or nucleic acid would actually form a droplet, and that's basically a way to compartmentalize reaction in a cytoplasm, right? So that's been observed in cells. Yeah, it's pretty interesting. Yeah. So instead of having a lipid bilayer as the compartment, or having a protein shell that some of the bacterial compartments use, this is basically all liquid, right? So there's no and they're very sensitive to some of the physical parameters, so temperature can change and the phase transition of these material, right?
Wow. Yeah. So I think we could move on to sort of the areas that are in the frontiers of synthetic biology now, and then that'll give us a good way to go on to how the two interlink.
Yeah. So for synthetic biology, I'll highlight a couple. Obviously, the field is 20 years old, and it's, a lot of it start out with engineering bacterial genetic circuits of bacterial in bacteria, and a lot of it has moved into mammalian cell synthetic biology. I think that's a very, very promising area, and some of the sort of advances that I particularly like are involve engineering of receptors on the membrane. A couple examples are the chimera antigen receptors, right? So that's these CAR T cells have been used in immunotherapy. And more recently, there's the development of SynNotch, right? So this is using notch receptors, so the extracellular side can be can have antibody-based extracellular domains that you can bind to ligand of interest. On the intracellular side, you have a essentially a transcription factor that can be cleaved up, and it can act on certain genetic circuits. So that's a great set of advancements. I also want to highlight, so my area of research in my lab is to work on cell-free expression in synthetic cell development. So sometimes I think about, when you think about embedding cells into extracellular matrix material, it doesn't have the living cells. It can be also these synthetic cells, right? And the way we define our synthetic cells are basically lipid bilayer, or it can be other materials. It's an encapsulated system, but it has we encapsulate cell-free expression reactions in there. So and cell-free reaction, for those that don't know, it's basically a biochemical system that can carry out transcription and translation in the absence of living cells. So that particular cell-free system has been used, for many, many years now. There's lots of interest in applying this to, say, biomanufacturing or other biotechnology applications. So what we do is we encapsulate that within our lipid bilayer vesicles, and we use that as a model for synthetic cells. So we're using it, to build different type of sense and response properties, so that they're very similar to what natural cells do, right? So natural cells sense a input and has an output. So in that way, I see a lot of synthetic biology tools that have been developed for bacteria and mammalian cells that are applicable for translating into a synthetic cell context. So I think those areas are, what I think is pretty exciting in the synthetic biology field.
Yeah, definitely. Sounds pretty cool. What is the link between the synthetic cells and the biomaterials? How can those two sort of merge together?
Yeah, I don't think a lot of people working on that particular area, but just happened that my lab is trying to do something now with embedding synthetic cells into a hydrogel, and we're and it just describe really briefly what that project is about. We've built synthetic cells that can respond to increased membrane tension. So these are mechanosensitive synthetic cells. So if you can deform the synthetic cells, you can actually open a channel on this synthetic cell, a mechanosensitive channel that will allow things to go in and out. So we started a study recently to embed this in a alginate hydrogel. It could be any other hydrogel. The idea is that eventually we can also embed cells in there, but our synthetic cells will be sensitive to the mechanical cue that we put in. It can be, it doesn't have to be mechanical. It can also be ligated. But the idea is that the synthetic cell would then release something to change the property of the alginate gel. In this case, we want to soften it so that cells can proliferate within this material. So I would say that's one example of how to integrate synthetic cells with bacteria. But I think most people have not been really doing that much integration between the two fields, and at least for synthetic cells itself, right? But that's just one example that we're trying right now.
And I'm curious, why is it, in your opinion, very sort of worthwhile to embed synthetic cells in hydrogels instead of a sort of engineering sort of cells that we already have?
Yeah, very good question. Yeah,
Yeah.
I mean, you can certainly do both. So
Yes.
one advantage of synthetic cells is that they're not living. They're basically, you can think of them as designer particles that you can engineer to serve a particular function, right? So the cells ultimately that we want to put in a gel could still be wild type. They're not programmed, to do anything, but we're programming the synthetic cell to sense and respond to something. And let's say we want the synthetic cell to be releasing something that would then affect the living cells, right? So I see synthetic cell as the middleman, right? Sort of, you have the gel, you have the external stimuli, and the synthetic cells are the sensor and responder, and those would then impact the living cells that are embedded in the in the material. But of course, I just talked about earlier that living materials, at least currently, a lot of people are doing this, who are doing this research, are embedding engineer cells already, right? So they're not really doing this with synthetic cells. It really depends on application, I guess. Yeah.
So is it, I'm just checking if I understand this correctly. Would you say that you view the synthetic cells as almost like of course you program them in a specific way, but when you build them, they're like a blank slate. So you don't have to necessarily worry about, especially if you're using cell-free technologies, worry about other sort of in cellular systems that could prevent your reactions from taking place.
Yeah, exactly. So I mean, yeah, exactly. I think the synthetic cells are something that you build in a laboratory, right? You build in a lab that you don't have to worry about. Like they can be independent of what your cells would do, but then you're sort of designing it with a specific purpose in mind, right? And then you embed that within the materials, along with cells, if you want the cells, and the cells here doesn't have to be, it can be engineering, it doesn't have to be engineered, right? Like, but you let the synthetic cell do the work, right?
Yeah. Right. Yeah. And how far is the research currently? What are you sort of specifically targeting?
Yeah, so not far at all right now. We actually just got a grant funded very recently, just a couple of weeks ago, actually. But the idea is to use it for tissue engineering purposes. Like the overall arching idea is, you'll be embedding cells in a material that will then eventually be, ideas to do this in vivo, obviously, eventually, right? But as cell grow, there's going to be increase in stresses within the materials, and want the material to be degraded. So we want to embed these synthetic cells in there to sort of modulate the stiffness of the material over time. And as the stress in the material builds up, the synthetic cells release, some molecule that will then degrade the gel, and sort of this will and allow the cell to continue to grow in that material. Yeah. So that's sort of the general idea. Yeah.
That's interesting. Can't wait to see how this progresses.
Yeah, I'm curious too. Yeah. But at least we gotta try it. Yeah, we at least got the funding to do it. Yeah.
Fine stuff. You also mentioned sort of synthetic receptors in the paper briefly. Could you talk a little bit about that?
Yeah, the synthetic receptors, I give a couple examples. One is this chimera antigen receptor that's used in immunotherapy. The other one is this Notch, right? So this Notch receptor, basically can sense, its native ligand, but it's really just, monitoring sort of cell adhesion. So people have used this to build like cell communication, right, between two cells. When the syn notch is binding to a ligand, it actually triggers a gene circuit. Like right now, a lot of times it's just a reporter gene. But this is very powerful because you can couple this to other type of output if you want. I think there are people that are, creating different type of outputs out there. And I think that's just the general concept of engineering a receptor that can sense a particular ligand, right? Like a cancer biomarker, for instance, right? Yeah.
Okay. It seems like there are so many options of like additives that you could have in the biomaterials. Like, where do you begin to even sort of process all that information.
So additive in what way?
So like,
sort of this?
I guess or like you can engineer things in so many different ways. Like you can have these different types of synthetic receptors. You can have, I guess, different cells that are doing different things. It just, seems like a lot.
Yeah, absolutely. Yeah. So I see where you're getting. Yeah. It's a good question, fair question. I don't know where one would start, but I suppose, depending on the application you're after, if you're looking at in a biomedical space of, targeting a particular type of cancer, you look for what type of biomarkers are upregulated, and then you ask yourself, how do you want to kill it, right? So there are different ways of killing a cell. I think once you have the receptor, engineer, then you look at what is the output gene you want to trigger, and do you want to just report that the cancer cell there? If that's the case, would you just need a fluorescent reporter gene, or do you want to have a light base reporter gene? So you have to sort of look at that sort of designing your receptor based on that, your system based on those information, right? What you're trying to do.
Yeah.
And so do you see sort of the use of maybe, well, I'm not sure if this could be applied to research, but do you see, the use of maybe machine learning or some form of automation in even like the the design of biomaterials?
So design, so definitely there's been a lot, going on in that space. So, I'm not an expert in, automation or machine learning, but we highlighted in our perspective that, both, machine learning and automation will, impact synthetic biology and biomaterials research. And I think, let me just sort of spell out why that's the case. And I think we already see this, being practiced, right? So you certainly would not, so the synthetic biology is built on this, design, build, test cycle, right? Like designing something or when you need to test a library, yes, you can have people that just do the pipetting in brute force manner, and that can only get us so far. So we had a couple jokes in our perspective, and I hope people take that well. But it is not easy if you ended up, say you want to test hundreds of sample, right, in a library of conditions. This is where robotic system actually is very, very helpful. And we know that this is in practice, people do use that, of course, whether you have access to a robot or not, it's a different story. But there's definitely that aspect of implementation. Now, machine learning can definitely help in identifying, so for biomaterial, for instance, you can help identifying, features that underpin a particular performance in the material, right? So as you design, let's say you have a material library, you design an assay for surveying the properties. Machine learning can help you make predictions as well as identifying the features that give rise to that particular thing. So one example of this is there's a study that show anti-fouling hydrogels. So hydrogels that basically things don't stick to it. It turns out that they discover a new composition that is non-intuitive at all, and that discovery can only be made by doing a certain set of experiments and then using machine learning to help make prediction, which can then be tested to sort of figure out, okay, this particular composition, because like you said earlier, there's a huge amount of space in terms of the design for materials, right? The combination is huge, but I think that is where high throughput screening, robotics, and machine learning can aid in that process. And similarly, synthetic biology has greatly benefit from some of those approaches as well. Yeah.
Could you?
How much of
Sorry, go ahead.
How do you use machine learning or automation in your lab? How far has it come?
So I'll say not in my lab. Not in my lab. We have not actually. Well, at least not in the automation part. I've been for a while wanted to get a instrument that's basically on the order of more than it's a large amount of money to do sort of droplet acoustic based droplet generation, so they can pipette very, very small volume.
Is that part of sort of a microfluidic machinery, or is that a
No, so we
separate?
do use microfluidics, and that doesn't that can generate synthetic cells in a high throughput manner, but in terms of screening, like in terms of having different compositions, microfluidic doesn't really help us there. What I'm referring to is there's a machine, not trying to advertise it too much, but basically there's a machine that can eject very small volume of liquid by using acoustic energy, and that machine can do high throughput pipetting basically, right? So that you can sample compositions over a wide range of conditions. But we don't do that. I mean, there are obviously robotics, robots that do pipetting, and I think there are, wealthy, sort of well-equipped labs may have that, or facilities may have it. In terms of machine learning, we're not using it in my own research. Have a pending grant on trying to look at peptide vesicles. So, just give you a brief idea. So, all the membranes are made of lipids. These natural lipids natural membranes are made of phospholipids, and there's this idea of using amphiphilic peptides as a way to build membranes. So, and it does exist, and you can actually take a amphiphilic peptide that will self-assemble into a bilayer. So when it comes to peptide, the advantage is that you can make it genetically encoded. Now, peptide also has a huge peptide space, right? Because you have amino acids that, there are 20 type of amino acids, and you get different lengths. So we're trying to use machine learning. If they're going to get funded, we're going to try to sample a few different type of peptides and then use machine learning to help guide that design, so we can discover new peptide sequences that will self-assemble into a peptide vesicle. So that's sort of a project we're hoping to work on in the future. Yeah.
Wow. Yeah, I never heard of that before.
Yeah.
Is that relatively new, or is it something that's been around a while?
Yeah, very good question. So I got inspired by it, relatively new. So peptide vesicles. There was a paper. These are probably in the 2018-ish time when a paper came out on basically these are elastin lipopeptides that oftentimes they form micelles, but it turns out they can also form a bilayer vesicle. And we had a paper last year that showed we can make these, giant elastin lipopeptide vesicles. So they're like basically lipid bilayer vesicle, except they're much stiffer, and will be a great compartment because they are, less fragile compared to lipid vesicle, but also genetic encoded, right? So the idea is you can use cell-free expression system to make these peptides. You can decorate the peptide with ligands, and essentially, instead of doing chemistry on lipids, you can do chemistry on DNA easily by encoding them with functionalities, right? So that's sort of the general idea.
Yeah, that makes sense.
Yeah.
Could you sort of bring together the ideas then that you've talked about in biomaterials and synthetic biology and how they can come together to bolster each other?
Yeah. Right. So maybe let me talk about sort of potential application, or I guess some of the examples out there. Maybe
Sure.
that's easier to sort of, So I talked about this concrete idea earlier. Maybe another example of this is a couple relatively recent work. One is on this bacterial cellulose based living materials that basically is a co-culture of yeast and bacterial cellulose producing bacteria. And then the approach basically is to take yeast strain that are engineered, and they can secrete enzymes into this bacterial cellulose. So bacterial cellulose is actually very widely used as a used as a material, and there are commercial products that uses bacterial cellulose, but this group basically embed yeast strains that are engineered to secrete enzymes into this material. So now they can autonomously grown as a catalytic materials, and actually they can modify the material properties of this bacterial cellulose. So if you can, and this is sort of why I talked about earlier, is this living material is an engineered organism, microorganism that is embedded in the material. So not just any normal yeast. But now if you can engineer this yeast, they can actually make this, cellular matrix that can sense and respond to different stimuli, like chemical or light, But you can also imagine, maybe this is not done yet, but it's in the work probably, that you can engineer yeast to be a biosensor that can detect certain chemicals or pathogens, and then basically have this be integrated within this bacteriocellulose material, right? So this is where I think, the synthetic biology part is this engineered yeast. The biomaterial is this bacteriocellulose, which is a natural natural material. I think this is where this intersection is starting to form, in this engineering living material, or this living material concept is very well illustrated. There's another study that, again, very recent, that is on fungal-based biomaterials. And basically, this involved the development of a living and self-healing fungal bacterial biocomposite that can form human-scale architectural objects. So they're not forming something small, and these are actually be materials that can be used in larger scale. And they also, what they did is they also incorporate engineered bacteria into this fungal biomaterials, and demonstrate they can have a sensory response mechanism, and also with stimuli responsive behavior. So I think those are kind of the examples that we're seeing now. And I think the opportunity is very, very large, because now the synthetic biology basically allows you to, engineer living organism, bacteria, yeast, and the biomaterial space is, was a static material. But now if you put in this living material in there, and that living material is going to sense and respond and have outputs, then you can start to have this bidirectional information flow between the materials and the living system that lives in there. Yeah.
How do you go about, say, so putting in the cells into this biomaterials? How do you grow a synthetic, like an engineered cell material? How do you go about making that?
Yeah. So I don't know the details of how they're made. At least the example I gave you with this, either bioconcrete or this fungal bacterial biocomposite, I think they're just mixing it. They have to have certain right conditions. So for the bioconcrete, I remember reading about it does require certain environmental conditions. Because I think one of them uses bacteria that requires high humidity, so you have to make these concrete in a high humidity environment. But they're literally just mixing them in a right condition, right? So if you go out to nature, I mean, there's coexisting organisms everywhere, right? So and for that kind of material, they're simply just mixing together, and they can put in the mold, they can shape it into a shape of the brick and whatnot. But if you go back to this other examples I talk about with embedding cells in a intracellular matrix material, that is also just mixing these cells together and let the gel polymerize around the cells. I mean, even though the scales are much different, the general idea is some type of is embedding the cells in the material, right?
Yeah, I'd be interested to sort of see the materials in person, like what they actually what they look like. And
that's right. Yeah, I think I've not seen them in person myself, but I remember looking up this bioconcrete example, or they're just like bricks. I think in the future it'd be pretty interesting. I mean, which is probably like building materials, you won't be able to tell. You can go to a home hardware store, and you will buy them as advertised as living materials, right?
Yeah.
We'll have, like, instead of greenwashing, we'll have material washing.
Exactly.
This is a biocomposite. It's like, is it really?
Yeah, exactly. I think this is all possible. I mean, this will be likely the future, right? And you can buy materials that are either just the static material, or materials with certain cells embedded in there that will have a certain property, self-healing, for instance. If the brick fractures, this is going to heal itself, right? Yeah, it'll be very cool. Yeah. Our houses will all be built by by these materials in the future, right? Yeah.
I think that might be a great place to end, unless you have anything you think we've missed.
No,
I have one more question.
Oh, sure. Yeah.
Can you hear me, actually?
Yeah,
yes, I do have one more question.
Yeah.
Okay, that's great. In your own work with these synthetic cells embedded in the hydrogel, I just wonder what sort of as you're sort of looking out at your research that you're about to do, a lot of research, what would you say are the main, I guess, sort of obstacles that you expect to face?
Sure. Oh yeah, yeah. No, that's a great one. So, they're planning for synthetic cells, but yeah. So I think for synthetic cells, I mean it's a relatively niche field within synthetic biology. It's one of those things that are much harder to do than one thinks. I got inspired by doing this because I think anything that cell can do, I want to be able to rebuild that. That's sort of my motivation. But as I got into this more and more, when you put a lipid bilayer around molecules, and we have to keep in mind these lipid molecules are actually pretty fragile. So one of the unknown is this, is that you build these synthetic cells that have very, very short lifetime, right? So that they're just not robust enough. But interestingly, my student have actually been able to demonstrate recently, since we got this project funded, that we can actually embed these, synthetic cells in hydrogels, and they can live in the sense that they are intact, over multiple days, up to a week or even longer. So this is promising. But I think in terms of the other challenges that we foresee is, we know what happens is when you encapsulate these cell-free expression system, it's not just the lifetime of the shell, the lipid bilayer itself, but also the machinery inside. So living cells have the ability to regenerate themselves, right? If you just feed them with food, they can live. Whereas synthetic cells, because they are essentially a system that will go with the equilibrium, you feed them with, you give them energy, right? In the sense of, nucleotide, they will run out. Of course, people are putting, the nutrient outside as well for them to come in and to sustain that life. But there's definitely a finite amount of life for this synthetic cells. I think that's going to be one major challenge. Of course, if you are going to build something that's terminal, that it doesn't need to continuously make new proteins, I think that's okay. But if you need something that is going to have to keep on maintaining that protein productions or whatnot, that this will be a huge limitation. Sort of metabolism of synthetic cell, I think, yeah, it's a huge hurdle, right?
I mean, yeah, I guess for wounds, so sort of for applications such as wound patches, they would be a less of a problem than sort of for longer term applications.
Yeah. If you need something that is, and I think, the conventional materials do just fine with like wound patches, things like that, of just having a material that doesn't need to, continually monitor the external stimuli, right? Like, most of the material we use in our day-to-day life is, I would say, non-responsive. They kind of just, they have a purpose, they're made for a purpose, and use them, and that's it, right? And of course, you will select the material based on what use you want to have. But once you have this living material, the idea is that it can start to, react to the environment, and then be able to have some, programmability that we talked about earlier, right? So be able to change its behavior or change its properties based on the conditions. Yeah.
Okay.
Thank
you so much. Thank you.
Sorry, one more, a very short one. Is there any sort of places that, like communities or like a society or anything, where someone could stay updated with this area of research?
Oh wow, yeah. I mean, there are a lot of so I affiliate myself more with the synthetic biology field, and there are lots of synthetic biology meetings now, or society meetings that have synthetic biology as a topic. Similarly, there's also biomaterials, meetings, and I think this might be a good way to end our conversation here. Is as we noted in our perspective article, I brought these two groups together, the most either synthetic biologists or biomaterial scientists. These two fields have grown tremendously over the past years, and mostly independently. I haven't know a bit about both, but I think it's and then some of the PIs also had that sort of cross knowledge between the two fields, but really they haven't been that integrated. The living material now is the area that really brings the two together. I'm not sure there's a dedicated meeting to just living materials yet, but certainly within individual societies, there would be topics, on this. I mean, maybe in the next few years, we may have dedicated meetings to that's at the interface of synthetic biology and biomaterials. Who knows, right?